Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. Thank you for joining us today. It is now my pleasure to introduce our speaker, Quincy Mehta of Unchained Labs.
Hi everyone, thanks for joining us today. Exchange your buffer exchange. Many of you have seen a washboard similar to what I'm showing here on the left. It's a very manual process for washing clothing, right? You can only use one article at a time. You have to physically wash up and down, put it through the rolling pin to dry it, and wring it, and repeat this process. But then not too long ago, the washing machine came out. As we can see, the nice front loader on the right here. Super easy to use. All you need to do is press a few buttons, load your clothing or sample, and add soap and hit the start button and away you go. What if there was a better process for buffer exchange? As we know now, buffer exchange is manual, much like the washboard on the left. Not only is it manual, but it takes a ton of hands-on time, right? First, you have to make your buffers. What that means is you're going to need to take your solid components, mix it into water, mix those stock components all together, and then you're going to stick a pH probe into the stock buffer solution. Is that pH the right pH? If it's not, you're going to add acid or base, titrate to your desired pH, and now you have your working buffers. But that's only the buffer preparation portion. Once you have the buffer preparation portion complete, you actually have to do buffer exchange. And as we see here, if you're using dialysis cassettes, you'll do buffer exchange. You need a lot of buffers to do that. But even after the buffer exchange completes, if you want to concentrate, you'll need to use spin filters or something similar at the end. And while it's not too bad for one sample, what happens when we have to do it for 1, 2, 3, even 12 different samples? We have 12 different formulations. If we're working on multiple projects, it just starts to increase exponentially. And this starts to become a real time suck. Buffer exchange downright sucks. I mean, look at this. On the left, we have a bioreactor that's automated. TFF, that's automated. A purification system's also automated. Why is it that buffer exchange and this concentration step at the end are still not automated? This takes an incredible amount of hands-on time and really slows down the drug formulation development process. If you're thinking to yourself it's a major bottleneck, you're not alone. As we can see here from our customers and other scientists out there from KBI, Glenmark, Genentech, and Pfizer, they all think it's a major time suck. But that's perhaps not even the biggest issue. What it's really doing is prohibiting people from getting to the best formulation. And while that can be consequential in the drug development process, even once you get further down the road, it can be an even bigger problem, right? Once we try to scale up to large-scale bioprocessing and reactors, if there's any sort of major production error when we're trying to do manufacturing, that can cost a company millions. So let's take a look at what's being used today. Currently, there's dialysis cassettes, many of you are familiar with, so slidolyzers, D-tubes, snakeskins. There's centrifugal filters, so most commonly people know Amicon and Paul. There's also solid phase extraction. I think most people know Zeba columns. Let's discuss dialysis. So many of you are familiar with this process, but one of the worst parts of doing dialysis is the liters and liters of buffer it takes to do dialysis. Not to mention, at the end, every sample needs to be concentrated. You can't do this within dialysis. On top of that, we move to centrifugal filters. Centrifugal filters also don't work very well as they require a ton of hands-on time, and once we get to the end, typically the mass recovery is going to be dismal, especially as we get to those highly concentrated formulations. Next, solid phase extraction. We also have issues with solid phase extraction, right? Because this is going to dilute our samples and there's no concentration step at the end. And I'm sure most of you are working with highly concentrated proteins, probably monoclonal antibodies, fusion proteins, or something of that nature. Solid phase extraction will not allow for concentrating at the end either. With that said, Unchained Labs wanted to take a different approach to this. We thought to ourselves, how can we automate buffer exchange? Well, we provided 3 different solutions. From left to right, we have the free slate option, Junior, and Grunt. Free slate and Junior are both going to be plate-based approach. They use our proprietary consumable known as Unfilter, which is a 96-well plate. What you'll notice with the free slate system is it's quite large, even though we made the image small. It's quite bigger than Junior and Grunt. What's cool about the free slate system is you can do multiple proteins, each in a microliter fashion. It's a fully automated buffer preparation and buffer exchange process, and you have the ability to integrate third-party analytics. On the left, you can see the carousel here, and this allows for the joining of multiple free slate systems together to allow those third-party analytics to be directly integrated. Next, we have Junior, which is also going to be the plate-based approach using Unfilter. This is going to be multiple proteins as well. The difference is going to be that the Junior is a dedicated buffer exchange system. You don't have the ability to directly integrate your third-party analytics. You have to do it virtually, so via computer, and the buffers will not be prepared on deck. Next, Grunt, which we'll be talking about more today, uses what we call the funnel-based approach. Funnel is a proprietary filtration cup, which I'll get into more later. Each of these samples is going to be 1 to 8 milliliters. The buffer prep and buffer exchange is all done on a single deck, just in a different form factor than the free slate system. And one other thing is there's no directly integrated analytics into Grunt. To review, let's see how Unchained Labs compares to other manual competition. With Unchained Labs' automated buffer exchange, you don't have to give any sacrifices. You have the ability to have a small amount of hands-on time, ability to have high mass recovery, an automated concentration step at the end, preparation of your buffers on the deck, as well as the ability to handle highly concentrated molecules. However, with dialysis, typically you only get the benefit of high recovery. Amicon, while you can concentrate, it's going to usually, once you go to increasing concentration, it's going to falter. The salting columns also work because they're a low amount of hands-on time. However, it's not going to handle highly concentrated molecules well, and there's no integrated concentration step. Why are people buffer exchanging? Typically, people are buffer exchanging for a variety of reasons. For example, people are doing it for analytics. They're doing it for sample preparation or characterization. After creating different formulations, they want to analyze them. In addition, people might be doing cleanup if you're going to run the samples on mass spec afterwards. In addition, people are also primarily using buffer exchange for formulation development. One of the primary applications for Grunt is formulation development. As we see here, at the beginning of the process, you have purification skids or TFF skids that are all automated. For example, the active purification system by GE that's automated. And then people are going to take these, do buffer exchange, and run them on analytics. As we see with the analytics, these are all high throughput or highly automated. So why is it that right now we're still doing buffer exchange with manual arduous processes like dialysis cassettes and Amicon spin filters? There's got to be a better way. With that, Unchained Labs created Grunt, the first automated buffer prep and buffer exchange benchtop system. The beauty of Grunt is it only takes less than 1 hour of hands-on time. You can prepare your working formulation buffers on the deck. Grunt has the ability to buffer exchange and concentrate samples all in an automated fashion. Grunt will produce 12 different or the same formulations, all at a final volume of 1 to 8 milliliters. Grunt can also process highly concentrated molecules up to 200 milligrams per milliliter. So far on Grunt, we've ran a variety of molecules, specifically fusion proteins, monoclonal antibodies, and enzymes. So let's take a high view approach and take a look at how Grunt actually works. When you first start a run with Grunt, it prepares the buffers. What that means is Grunt will take up to 12 weak acid conjugate-based pairs, or buffer species if you're more familiar with that, combines them with up to 8 different stock excipient solutions. Then Grunt uses liquid handling and positive displacement tips to mix these solutions together to provide you working buffers. Once Grunt has made these working buffers, it'll use its Mettler-Toledo inline pH probe to take a pH measurement of each of these buffers, and if the user so desires, they have the ability to alter the pH to a different desired pH. Grunt uses a strong acid and strong base that are on deck to automate this process and adjust the pH to the user-specified number. Once Grunt has adjusted the pH and finished mixing the working buffers, you're provided with your 12 formulation buffers, which will be used for buffer exchange. Once the buffers are made, Grunt moves over to buffer exchange. Taking a look at the buffer exchange process, Grunt takes the starting protein sample, will concentrate it down, add new buffer, and repeat this process until 96% exchange is reached. You notice the 96% exchange we use on this chart here. However, Grunt actually has the ability to do anywhere from 90 to 99.5%. 96% is the default, but the user has the option to set the number to their desired percent exchange within the Grunt software. Once Grunt does this with one sample, it actually can do this with up to 12 different samples all in parallel, as we can see here. So to remind everyone, these can either be 12 different formulations, 12 of the same formulation, or any combination of formulations. This really gives the user a great amount of customizability with the system. As I alluded to earlier, we use a proprietary-based consumable known as funl. This is how Grunt's going to do buffer exchange, and this is integral to the way Grunt does buffer exchange. To tell you a little bit more about the funl is a proprietary consumable made of polypropylene. It has a 10 kilodalton molecular weight cutoff membrane made of regenerated cellulose. The final volume in each funnel is going to be anywhere to 1 to 8 milliliters. Once again, this is a user set parameter. How does Grunt actually do buffer exchange? If you follow the pointer here, Grunt will add the protein sample. It measures the volume. The way Grunt measures volume is actually a really cool approach. It uses fill times, so we pressurize each of those funnels up to 60 psi with nitrogen. Grunt measures how long that time takes, and from there we can extrapolate out how full the funnel is at any given time. Once Grunt has measured the volume, it applies additional pressure and a slight orbital mixing to pull through the old buffer, and then Grunt measures the volume again, adds new buffer, and repeats processes 3, 4, and 5 until we reach the desired percent exchange. From there, the user has the option to concentrate at the end if they so desire. Now that we know how Grunt looks, let's take a look at the inside of Grunt and see how the system actually looks. Taking a look inside the system, we see a really clean design. We see 3 main areas. On the left, number 1, we see the positive displacement tip rack. These are all Eppendorf PDTs similar to what you're already using in lab. 2 is the buffer preparation tray. This is what Grunt will use to make any of the initial stock buffer solutions, and then it'll make the working buffers towards the back of this deck. 3 is the buffer exchange module. So this is that 12-chamber module which will be loaded with funnels. We'll talk about that more on this next slide. The buffer exchange module is a 12-chamber system. Each of those chambers is going to be individually pressurized to 60 psi with nitrogen. Not only that, but each of these pressure chambers is individually controlled. What's cool about this is if there's ever any issue with a specific chamber, that won't catastrophize the whole, it won't end the whole experiment, right? Grunt software can isolate the well and continue on with the other ones. In addition, each of these chambers is going to be calibrated prior to each run, which ensures a very accurate volume measurement. To add another great feature, we wanted to temperature control this unit. The user has the option to set this module anywhere to 4 to 25 Celsius, and that module is controlled as a single unit. In addition to controlling the temperature during a buffer exchange run, we know Grunt might finish at night. Most people aren't going to want to go in to reclaim their samples. So what we did was we allowed the user to temperature control after buffer exchange. What's nice about this is the temperatures are set differently. So if you want to buffer exchange at a higher temperature to make the process go faster and then cool down once the buffer exchange is complete, you have the ability to customize that. Not only is the buffer exchange module temperature controlled, but we also know everyone's molecules, proteins are very sensitive. So we also temperature controlled the protein station, which is where the protein will sit during buffer preparation before it's placed into the pressure chamber for buffer exchange. As with the buffer exchange module, this is also temperature controlled between 4 and 25 C. Now that we know how the system operates at a high level, how it looks inside, let's take a look at the Grunt software. Grunt software was designed to be extremely easy and user-friendly. We wanted to make sure anyone in the lab could walk up to the system and use Grunt at the click of a button. Because dialysis and Amicon, spin filters, those are all simple processes. We know with automation there can be some scary things that come about, complex software, hard to use operation. So with Grunt, we really wanted to make it user-friendly and digestible. As you can see on the Grunt screen, we have 3 main areas: runs, buffer recipes, and solution inventory. When you're first using Grunt, you'll read this from right to left. Solution inventory is going to be any stock solutions you input to Grunt at any given time. Once you've added solutions to your solution inventory, you'll move over to the buffer recipe section. Once in the buffer recipe section, you'll take those stock solutions you already inputted, insert some numbers, and tell Grunt, this is the working buffer I want to make. So that'll become my formulation buffer. Once you've done your buffer recipes, you move over to the run section. So we say runs, Grunt run, but that's also just basically an experiment. But with Grunt, it's a run because that's the way we decided to name it. Once you have your buffer recipes inputted, you'll go over to the experiment screen, and you can take those buffers and do an actual run. Starting a run is as easy as clicking the Go to Runs button. Once you're brought to the Go to Runs button, you'll see 3 main areas: designed, completed, and templates. The designed area is any runs that have been started but not yet completed. So that's another great feature of the Grunt software. Say you start designing your experiment, but you have to go do something else. You can just save it and come back at any time. Once a run's completed, it'll go into the completed section. What's cool about the completed section is you can examine any post-run that happened. So any sort of information you want to gather, it's logged, which is great for tracking purposes. In addition, say you want to do a run multiple times for whatever reason. Either something went wrong, or you want to do a very similar run but maybe a different pH, a different formulation screen, anything like that. Or you want to do the run in duplicate or triplicate. You have the ability to template a run using the button down here, Save as Template. And what this will do is it'll automatically fill in it'll pre-fill in all the different areas of setting up the experiment or run, which will make it super easy to get started and take that hands-on time down to even less than an hour. So let's create a new run. If we click the Create a New Run button, we'll be brought to the following screen. Once again, we see it's super easy to use. All the user needs to do is insert the run name, so that's up to the user to decide what to call it. Run description, provide a little more information about the run, so if you have any questions that the run name didn't cover, you have a descriptor in case you want to look at it later. And you select the run type. Grunt has 3 different run modes. So you can do a full run, which means Grunt will take those stock solutions, prepare the working buffers, and then buffer exchange. Buffer only means Grunt will only make the working buffers. It won't actually do buffer exchange. Buffer exchange only means the user has made the working buffers offline, will put those into Grunt, and Grunt will only handle the buffer exchange portion of the process. Once the user has named their run, entered a run description, and selected the run type, they'll add their buffers. So adding buffers is as simple as clicking this blue button right here that says Add Buffer Recipes. And if we remember, the buffer recipes is that green section in the middle where we input those before we started the run. All we need to do is click the blue button, Add Buffer Recipe, and we can quickly add our desired buffers. Remember, the user can go anywhere from 1 to 12 buffers. Once the user has input their buffer recipes, they'll move over to the protein formulation section. Inputting protein formulation information is simple as well. All the user needs to do is enter their protein name, so that could either be the actual name of the molecule or a code name. They'll enter the initial concentration, the final concentration. So Grunt will use this number to tell if you want to do a concentration step at the end. And in addition, Grunt also has the ability to calculate the initial or final volume. What's cool about this is, say you have a limited initial stock. So what you'll do is, you'll tell Grunt you want to calculate the final volume because you're going to know what your initial volume is and know how you're limited. Or you can have Grunt calculate the initial volume. What this means is, say you're doing a specific analytic or doing a certain type of process after buffer exchange and you need a very specific amount of volume. You can tell Grunt, this is what I need at the end, and then Grunt will calculate what the input needs to be so that Grunt can buffer exchange and provide you your necessary output. This makes doing the run a breeze. Once you've input your protein information, you'll see all the different buffers you've added that you'd like to be made during this run. In this case, we only have one buffer. Here is also where you'll get to add in if you'd like to do a desired surfactant. After buffer exchange, Grunt spikes in a surfactant at the end. On the Grunt deck, so the buffer preparation tray, the user can add up to 6 different surfactants. Grunt will add one of those at the end of a buffer exchange run. Once your protein information has been input, you're brought to the instrument parameter screen. The instrument parameter screen is where the user really gets the opportunity to customize their Grunt run. Starting out, the buffer preparation, the user has the ability to decide if they want to do a 2 or 3 point pH calibration. Both these pH calibration processes are USP 791 compliant. The user has the ability to decide whichever pH standards they would like to use. And Grunt uses that Mettler-Toledo in-lab pH probe to complete the pH measurements as well as the titration. Not only do the user get to select the pH process, but the user also gets to select the buffer stirring speed. For most buffers, we recommend keeping it at medium, but say you're dealing with a highly viscous sample. You're definitely going to want a higher stirring speed to make sure your buffer is mixed appropriately. Those are the options the user gets to do for buffer preparation. Once moving down to buffer exchange, the user gets the option, as we mentioned before, to set the protein temperature anywhere from 4 to 25 C. They get to choose the buffer exchange temperature anywhere from 4 to 25 C as well. And this is where you'll get to choose that post-exchange temperature between 4 and 25 C. Not only that, but the user gets to customize the buffer exchange process Grunt is using. They have the option to set that desired target exchange percentage between 90 to 99.5%. They get to select how much of the buffer they'd like removed per cycle. And Grunt also does an orbital mixing feature so that the user can avoid any polarization on the membrane. The user has the option to program the duration of this orbital mixing process. You'll also notice I didn't mention it, but there's 2 different UFDF options. We have dilute and filter. Filter and refill is the standard way Grunt does buffer exchange. And that's what I described earlier, where Grunt takes the initial protein stock sample, concentrates it down, adds new buffer, and repeats this process until they reach the desired percentage exchange. However, we mentioned Grunt can handle highly viscous and concentrated molecules, up to 200 mg per mil. So what if I'm doing that? I know buffer exchange times can go quite far. Well, the beauty of this is that if Grunt uses the dilute and filter process, we can actually cut down on that time. The dilute and filter process is going to be similar but different. So what Grunt will do is take that initial protein stock, add it to the funnel, but then instead of concentrating down, Grunt will dilute the initial buffer with some of the working buffer and repeating this process until the desired percent exchange is reached. Once the user has selected their instrument parameters, they'll be brought to the shopping list. The analogy we like to make to the shopping list is it's like going grocery shopping. Much like a grocery list that will contain any items you need to do a recipe and do your cooking, Grunt will provide you a list of every consumable as well as every stock solution you need to input in Grunt to perform the experiment you'd like to accomplish. Not only that, but Grunt also has the ability for the user to input a lot number as well as an expiration date of these stock solutions. Many of you will be getting your stock solutions from essential repositories. So it's important that if you're doing this, you have all the tracking you need that would be essential to seeing if anything went wrong. Everything is tracked out so we can diagnose what happened. In addition to being able to track the lot number and expiration date, the user also has the buffer labware map. So in this case, we show the buffer labware map, but there's actually a map for every single thing you'll load into Grunt. What's cool about this is it'll tell you, if in the case Grunt's making a working buffer, it'll show you where that final working buffer will be. Or if it's a stock solution you're going to input, it'll tell you where exactly each stock solution needs to be inputted for Grunt to function successfully. Once the user has found all their required stock solutions and consumables, they'll move into the pre-run guide, which is where you'll actually load the system. The cool thing about the pre-run guide is it's just another check to make sure the user doesn't confuse Grunt by putting anything in the wrong spot. So what this is going to mean is Grunt's going to have you check off everything you need to input into the system. It's going to make you check things such as the KCl level for the pH probe or pH 7 standard, as well as other standards. It'll have you load all the consumables here. You'll also empty the waste, fill the water reservoir, and clean the instrument off. What the pre-run guide is it's a great way to check to make sure everything is set up and ready to run. Once again, making Grunt really usable by everyone in the lab, not just a dedicated automation person. Loading a Grunt is super easy as well. As we can see, these are the easy to remove racks. On the left, we have the positive displacement tray, and on the right, we have the buffer preparation tray. Number one is the 10 milliliter positive displacement tips. As I said before, these are Eppendorf tips, just industry standard. Grunt will use the 10 milliliter positive displacement tips primarily for everything, except there are situations where Grunt will use these 1 milliliter PDTs, which is number 5. The 1 milliliter PDTs will be used to add strong base, strong acid, as well as surfactant. As we don't want to overshoot when we're doing titration, so we want to do it very precisely, and the 1 milliliter positive displacement tip allows us to precisely add those desired components. Going from the back to the front, we see the magnetic gear track, and this will be what controls the stir bars in the working buffers, so we can mix those working buffers. And then moving towards the front, we have the stock solutions, which are number 4 here. Number 6 will be the stock excipient solutions. And then going left to right, we have strong acid, surfactants, pH standards, and strong base. What's cool about everything in this deck, all these solutions are customized to whatever run you're going to be doing. The only thing that always stays the same is the type of consumable. What's also cool about this is every consumable here is different. So there's no way you can misplace certain consumables in the wrong spot, as everything only has one specific fit. Once you load the buffer preparation tray and the positive displacement tips, you'll load the funnels. What we did with the funnels is make it easy to carry rack here that'll screw down into the module. As you can see, the screws here, 2 screws will screw into the buffer exchange module. The user will load their 12 funnels, and then they'll screw down the top portion. What's cool about this is once a run's done, it's super easy to remove this rack by unscrewing just 2 of the screws, and it has a nice carrying device here that you can take it over to a lab bench, pipette out your samples, and reclaim the samples. Once Grunt is all loaded and actually running, how do we know Grunt's doing what we want it to do? Well, we created this easy-to-use status screen. On the status screen, you'll see a bunch of bright colors, which is, once again, very nice, easy to use. We see the time to completion. So this time to completion, during the buffer preparation portion, it'll show how long it'll take to complete buffer preparation. Once buffer preparation is completed, it'll show an updated time to show how long it's going to take to complete buffer exchange. This updates in real time. Moving down, we see the buffer preparation for each channel. In this run, we're doing 4 channels. We can see what we wanted the target pH to be, so this is the user inputted number. And then we also have what the actual pH was. And this will update in real time as Grunt completes these working buffers. Once the working buffers are completed, Grunt will move over to buffer exchange, and it'll show how far in the process it is for each specific buffer exchange channel. In addition, the status screen also shows the current protein temperature as well as the buffer exchange temperature. One final feature of the status screen, which is really nice to have, is this gray text at the bottom here, where you see it says refilling buffer exchange filter cup number 1 with new buffer. What's great about this is at any time, Grunt will tell you exactly what it's doing. There's never a mystery what's going on. You don't have to worry if Grunt's faulted or anything's wrong, because you can check with a quick look at the screen what Grunt's doing. Once a run's complete, you'll be brought to the run report, which offers full traceability for that experiment. You notice such things as the instrument ID, run name, run description. Remember, those are user input. Who did the run? What type of run? Remember, Grunt has 3 different run modes: buffer exchange only, buffers only, and full run. It'll say how long the run took. It'll also catalog what the working buffers were, as well as what the target and actual pHs were, any of the different stock solutions you use, and as you go down, it'll also show what those desired final formulations were, as well as what the final volume is after the run for each of the specific channels. In addition, Grunt can easily be exported to a CSV, which is great for ELNs. By clicking the Export to CSV button, you can easily save into Excel. Or if you're using a hand lab notebook, you can easily print this off and put it into your lab notebook. In addition, the run log is another feature of Grunt software where it catalogs every specific action Grunt took. The great thing about the run log is if anything went wrong or you're wondering why something's taking so long, you can quickly diagnose by looking specifically into the run log what's going on. This makes operation of Grunt very easy. Now that we know how a Grunt looks, how it operates, and how easy it is to run, let's take a look at how Grunt compared to some traditional methods. One of the first experiments we wanted to run is we wanted to see how an automated buffer exchange approach looked to a traditional method. What we did was we gathered molecules, in this case 3 monoclonal antibodies and an enzyme, all from biopharma partners we worked with to help develop Grunt. What we did was run these molecules on Grunt using buffer exchange. We ran it on spin filters, in this case we use Amicon, and we also buffer exchange on dialysis using Thermo Fisher slidealizers. Once we ran these 4 molecules, once we buffer exchanged these 4 molecules on these 3 different processes, we wanted to see mass recovery afterwards, pH accuracy, as well as protein quality. We checked SEC and DLS. The SEC and DLS were all done immediately after exchange. What we found was Grunt easily hit the target pH. We'll notice here for monoclonal antibody alpha, monoclonal antibody beta, and monoclonal antibody gamma and the enzyme, Grunt was within 0.1 pH units, which is really accurate. We also see Amicon was within 0.1 pH units. Dialysis was a little further off, though, sometimes reaching as high as 0.2 to 0.4 pH units off from the target. In addition to hitting pH accurately, Grunt also gave great mass recovery. As we notice here, Grunt either performed better than or at the same as the competition. With monoclonal antibody alpha, Grunt hit 96.1% mass recovery, which was higher than both Amicon and dialysis. For monoclonal antibody beta, Grunt hit higher than Amicon and dialysis again. Monoclonal antibody gamma, Grunt was just slightly under dialysis by 0.02%, and once again higher than Amicon. And with the therapeutic enzyme, Grunt hit higher in mass recovery than both Amicon and the Thermo Fisher slidealizers. Now that we know that Grunt hit accurate pH targets as well as had high mass recovery, what was the protein quality like? Well, if we look at the chromatograms for all these, we see a similar story. We see a great monomer peak around 17 minutes for monoclonal antibody alpha, not much degradation in the form of aggregates or fragments, and then we can see the buffer peak at the end. Same with SEC, or with the chromatogram for monoclonal antibody beta. We see the aggregate peak a little bigger. Once again, a strong monomer peak around 18 minutes, and we also see the fragment peak and then the buffer peak. For monoclonal antibody gamma, we see a strong monomer peak, small amount of aggregates, small amount of fragments, and the buffer. Again, the SEC, so the chromatogram for the enzyme, is also going to be similar. What we can gather from this is Grunt's clearly processing these molecules similar to Amicon and dialysis, and is taking care to not cause any degradation during the buffer exchange process. One other thing we wanted to check was DLS. Once again, with DLS, similar to the SEC, we see a similar story. We see strong monomer peaks for the monoclonal antibody alpha, beta, gamma, and the enzyme. We see that throughout for both Grunt, Amicon, and the Thermo Fisher slidolizers. Once again, low polydispersity and a high monomer content peak. As we mentioned previously, one of the great selling features for Grunt is the ability to handle these highly concentrated molecules. As we know, the industry is always trying to get to what is the highest concentration we can hit so we can deliver the least amount of sample. So we wanted to provide something that's going to really fit into this, where the industry is moving. So what we did was, once again, we ran Grunt dialysis. We used Thermo Fisher slidolizers and Amicon. We ran 2 different highly concentrated IgG molecules. We measured the hands-on time and the total time. To reiterate the process with Grunt, to do buffer preparation and buffer exchange, the user would program a run, input their stock solutions, load Grunt, start, and then remove the exchange and concentrated protein. With dialysis, it's going to be the traditional process. You prepare tons and tons of those working buffers, fill beakers with buffers, add the cassettes, dialyze, add new buffer, repeat that process over the next 24 to 48 hours. Amicon, it's going to be similar, adding buffer, washing the spin columns, prepping and adding the protein, spinning, adding new buffer, and you have to do more and more cycles as the protein gets more highly concentrated. Taking a look at the results, Grunt got it done, even with a 200 mg per ml protein concentration. We'll notice here the hands-on time with Grunt was literally only an hour, whereas with dialysis, scientists had to dedicate 34 hours of hands-on time, and Amicon 91 hours of hands-on time. With Amicon, that means it took over a week of a full-time employee to do that buffer exchange. Not only was the hands-on time minimal with Grunt, but also look at that processing time. With Grunt, it's only 3.8 days, dialysis nearly double at 7.5, and Amicon off the charts with 13.4 days. Grunt really hits it home here, though, with the mass recovery. We'll see Grunt is comparable to dialysis at both 87% mass recovery, Amicon coming in at a low 46%. In addition, Grunt was able to hit the final volume, 7.8 milliliters, almost right on target to the 8 milliliters. With dialysis and Amicon, both of those are uncontrolled. Now that we looked at a 200 mg per ml IgG, we want to look at 150 mg per ml. Once again, we see a similar story. Grunt, only 1 hour of hands-on time. Dialysis, 34 hours. That's that fixed time, as you just let the dialysis cassettes sit and spin in the buffers. Amicon coming in at a high 72.4 hours again. Once again, the processing time is incredible. Grunt only took 2.8 days, dialysis 7.5 days, Amicon 9.1 days. Once again, Grunt and dialysis, clearly the winners of mass recovery here. Grunt coming in slightly under dialysis, but once again, great recovery. Amicon at 62%. To summarize, Grunt handled both of these highly concentrated molecules really easily with a small amount of hands-on time, great processing time, and high mass recovery. One final study we did was we had another biopharma customer who provided us with an extremely concentrated monoclonal antibody. So this is 200 mg per ml, and this is a therapeutic monoclonal antibody. We did a similar process where we measured hands-on time, total time, and mass recovery. To remind everyone, it's the same processes we used before. Grunt, you program the run, prepare the stock buffer solutions, load the system, hit start, walk away, and grab those exchange proteins and concentrated samples. With spin filters like Amicon, we prepare the working buffers, wash the spin filters, prepare the protein and load, spin, add new buffer, and repeat this process until we reach the buffer exchange. How did Grunt fare towards Amicon? Once again, Grunt delivered on time. In this case, though, you'll notice Grunt actually took 2 hours of hands-on time instead of 1 hour. And that's because this molecule was highly viscous, so pipetting out the final solution at the end took some time. Amicon coming in at 77 hours. We will notice here, though, that Amicon was able to process the molecule slightly faster than Grunt. However, once we start to look at mass recovery, we see a different story. Despite Amicon processing 0.2 days quicker, look at the mass recovery differences. 88% for Grunt and only 58% for Amicon. I think it's an easy decision what you would choose here in this case. We know these samples are incredibly expensive, and losing any amount of this initial protein stock can be catastrophic. Not only did Grunt have high recovery, but Grunt also was almost exactly on target with the final volume coming in at 8.02 milliliters. With Amicon, we had to dilute that target at the end. To summarize, with Grunt, you never have to buffer exchange alone again. Grunt is literally only going to take less than 1 hour of hands-on time to have you be provided with your prepared working buffers and buffer exchange sample. All you need to do is 1 hour of your time. Grunt excels against dialysis and Amicon, right? In terms of hands-on time, it's always going to be better because it's only going to take 1 hour. And in terms of mass recovery, against Amicon, Grunt is a clear winner. Grunt can also has the ability to process highly concentrated molecules, which Amicon is going to falter with. Not only that, but you actually have the ability to control your end result, right? The user will input that initial protein stock concentration. You have the ability to put what you want the final concentration to be, but you also have the ability to control what the final volume is as well. So now it's time to get yourself a Grunt so you can sit back at the beach, have a beer, and really just enjoy life while Grunt does the nitty-gritty buffer preparation and buffer exchange process.
Thank you, Quincy. Are you ready to answer some questions? We have a few from the audience.
I think I am.
Okay. Can the funnel be sterilized?
So at this time, funnels aren't sterilized. The instrument itself isn't in a sterile environment, so no.
And what is the maximum volume of stock solution Grunt can store?
So it depends on which solution it is. So for the working buffers, those are 125 milliliter, basically square Nalgene beaker type bottles. With the excipients, it's going to be slightly less. The nice thing is, though, basically Grunt software will tell you what the minimum amounts it needs to complete a run, but you can just kind of fill them up as high as you want. So that really, it's nice to be able to, you only need to know the minimum.
And are there automatic flush and clearing processes to ensure no harmful material is left in the system?
Yeah, so what the user has the ability to do is they can flush the system after buffer exchange. There's a protocol within the software that all they need to do is click a button that says flush buffer exchange module. And what Grunt will do is it'll flush a couple liters of water through the system just to clean everything out.
Is it possible to have a Mettler-Toledo in-lab Pro pH probe to prevent buffer interference in the measurement?
So it actually is a Mettler-Toledo in-lab pH probe. So the pH probe will actually be washed in between every time the pH probe enters either the initially mixed together stock solutions or even once it's doing the titration. After the probe goes into a working buffer, it will always be washed and dried in between each of those processes.
Is the Grunt software compatible with CFR 21 data integrity standards?
Right now it is not.
Are all the liquid handling components single use? And if so, what components are?
Yeah, so the nice thing is basically Grunt will have you load in however many positive displacement tips it needs to do buffer preparation, buffer exchange. That is to say each tip will only touch one specific liquid. There'll never be any tip touching more than one liquid.
Is there anything that needs to be cleaned and sterilized between runs?
Yeah, so it doesn't need to be sterilized, but we do recommend cleaning everything with dilute ethanol, IPA, or bleach, because what will happen is if there's any sort of drips of these stock buffer components, right, everyone knows they're extremely high concentration salts, so you will start to see some crust buildup, but what's easy to do is after each run, just doing a quick wipe down.
And do you find that Grunt significantly saves on time as well as labor compared with something like a NAPD salt column?
Yeah, I mean, right, the hands-on time is significant with Grunt, like how little it takes, right? The system is literally in my opinion, anyone can walk up to it after being shown how to use it once, set it up in less than an hour. I mean, if you already have your stock solutions made, I would safely say you can get a run going in 15 minutes.
And are there any funnel filter cutoffs higher than 10 kilodaltons?
So currently we only offer one membrane cutoff, which is 10 kilodaltons. So primarily, right, this is going to be a great application fit for monoclonal antibodies, but anything that won't go past the 10 kilodalton cutoff will work fine on Grunt.
Okay, so there's a question about the SEC chromatogram of the MAB gamma. You know,
yeah, I mean, so the thing is, yeah, definitely. So looking at the gamma and enzyme, yeah, I think looking at it now, it does look like it was a copy and paste issue. So we can correct that when those slides are released.
Okay. And how is Grunt in terms of solvent compatibility?
I think I would need more information on what they're getting at there. My contact info is at the end of this slideshow, so please reach out to us and we're happy to give you more information. I mean, I'd love to chat further and see what you're getting at.
Okay. Is it possible to increase the temperature for stress tests?
Currently, the temperature of Grunt is only 4 to 25 C, so it's going to limit stress testing capability. Yeah, so as of now, it's 4 to 25 C. But that would be a nice feature to have, the ability to get to like a 40 degree C temperature, so you can do that type of testing.
And what types of molecules have been run on Grunt?
So we've ran a variety of monoclonal antibodies, fusion proteins, and enzymes. And those have all been in a variety of concentrations, right? If we look back at some of the slides, one second. We see, so with that first study, we were running molecules as unconcentrated as 1 mg per mil, and we've also gone as high as 200 mg per mil. And we've run everything in between, so like 5, 10, 20 mg per mil, 75. So anything in that 1 to 200 mg per mil range should run no problem.
Can you run less than 12 funnels at a time?
You can. So the user, right, when they're in that run creation page, they have the option to go anywhere from 1 to 12 funnels. So not only are you able to do different formulations, if they don't want to run on 12, they could do 1, 6. The combinations are endless.
And we've got a question about, can the system control the final sample concentration?
So I mean, what it's going to do is, in the sense that if I input 10 mg per mil in that initial concentration area and input then 20 mg per mil in the final concentration area, it'll control it in the sense that assuming, right, you have full mass recovery, it'll be able to control it in that sense that it can alter the volumes and concentration accordingly.
Do you have other MWCO for the funnel?
Currently, it's only the 10 kilodalton molecular weight cutoff, and that's a regenerated cellulose membrane.
And what type of pH electrode does Grunt use, and does it measure the final sample pH?
Mettler-Toledo in-lab pH probe.
Okay. Well, thank you, Quincy. I think that's all the questions we have. The recorded version of the webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with a direct link. So we look forward to having you join us at future BioProcess International Ask the Expert webcasts. We have a full lineup scheduled for the duration of the year. Look for those announcements in your inbox. Thanks again, Quincy.
Yep, thank you. I hope everyone enjoyed it today.
Thanks. Great, thanks. Goodbye.
Bye.